Hello everyone and welcome. Thank you for joining us today for a practical guide to automating laboratory workflows. I'm Sarah Mackey and I lead the sales and marketing team here at Hudson Lab Automation, and I'll be your moderator for today's. We're very excited to have you here with us for this webinar. Before we get started, I just wanted to give you a few quick housekeeping notes. All attendees are muted throughout the presentation. We'll open things up for questions during the live Q&A at the end. Please feel free to submit your questions at any time using the Q&A panel, and we'll address as many as we can. Today's session will be designed to be practical, real world, and grounded in how labs actually work, not automation theory. I'm pleased to be joined today by two experts who bring complementary perspectives across science, instrumentation, and automation. First, I'd like to introduce Dr. Richard Cuthbert. He's the Global Product Manager for Cell Biology at Bio-Rad Laboratories. Richard brings more than 20 years of experience in flow and cell biology, spanning academic research, applied cytometry, and global product leadership. At Bio-Rad, he leads strategy and innovation for flow cytometry and antibody technologies with a strong focus on screening and high throughput applications. Joining Richard is Andrew Witschi. He's our Chief Technology Officer here at Hudson Lab Automation. He has nearly 2 decades of experience in laboratory automation and has played a central role in shaping Hudson's modular automation platform. He leads innovation across software, robotics, and workflow orchestration with a focus on making automation scalable and accessible for modern laboratories. He's been involved in or directly led over 100 automation integration projects for biotech and academic researchers. Before we dive in, a quick brief overview of the two organizations behind today's discussion. Bio-Rad has been a leader in life science and research and clinical diagnostics since 1952. From research to diagnostic, Bio-Rad's mission is to advance science and help labs transform complex biological questions into meaningful outcomes. Hudson, founded in 1983, has decades of experience in liquid handling and scientific workflow automation. We help research biopharma and diagnostic teams transition from manual processes to scalable, flexible automation. So Hudson and Bio-Rad have collaborated closely to streamline and simplify the automation of flow cytometry, qPCR, and PCR workflows, enabling labs to scale automation in a practical, stepwise way. Today's webinar is about practical automation. Rather than viewing automation as a single large investment, we'll explore how labs start with a single automated task, expand incrementally as needs grow, and ultimately scale towards reliable walk-away workflows when it makes sense scientifically in operation. We'll discuss why labs actually automate, beyond just throughput, how to decide what to automate and when, how flow and qPCR fit into broader automated workflows, and what to look for in automation-ready instruments and platforms. To kick things off, I'm going to hand it over to Andrew, who will frame how labs should think about automation, how to approach it in a modular, scalable way. And over to you, Andrew.
Thank you, Sarah. And thank you, everyone, for joining us today. I'm looking forward to the opportunity to share some of my experience with you. I wanted to start out today kind of looking at some of the real drivers for why labs automate. Diving into that, I think it's important to start with a question of what is automation. When we talk to customers and other partners in our industry about this, everybody has a bit different view on it. Some people think of pipetting. Some people think of plate reading. Some people think of big systems. Some people think of unattended. Some people think of things that they interact with every day. And there's a lot of customers out there that are going from handheld pipetting and wondering what is the next step, right? So I think one of the key points here, which is a little bit ironic in a webinar about automation, is that we all really don't have a consistent definition of what automation is. It's a really big picture word that spans a lot of different contexts and a lot of different scales. And I think the key finding here is looking at automation is really just a tool. It's not an answer. It's a tool that you use, and it solves a variety of different problems. when we work with customers, we find a lot of different reasons that labs actually automate. I think I'll hit one point here. When I'm working with our internal teams at Hudson, really try to strive to focus on data, right? The problem that we're trying to solve in our industry is to come up with solutions, right? And in the process of doing research is about creating data, creating quality data, and ideally creating that data faster than the competition. So when we look at the primary drivers for why labs automate, a lot of that is rooted in the quality of the data, right? That's reproducibility. It's CV, whether or not a task is done. It's reducing error rate. Sometimes in data, it's about the traceability of the data, right? And then there's a second tier that really starts to interact with users. And when we start talking about users, we look at that somebody sitting there working with an instrument can't be sitting there in front of it all day, every day. So you get a microplate reader or flow cytometer, and you got 24 hours in a day, and typically most of those instruments are only going to be running for a quarter of the day or less, just because somebody's not sitting in front of the instrument, or it's tied to the schedules of when users are in the lab. And then critically, there are the drivers of once you've met the requirements of generating high quality data and doing it in a way that works for your lab, then you start talking about how can we create that data quicker, right? And that's where throughput starts to become a more significant factor. So I hit a couple more points there where throughput is not the ROI, right? If we really want to focus on value, the value of this is really going to depend on what research is being done. Some research is more driven by time, where some research is more driven by volume. I mean volume of data points, not volume of liquids, obviously. So every lab is different, every process is different, and when you want to look at automation, the key is not to have a fixed answer in your mind, but to really look at what you're doing and what support you have in your organization, and try to find a solution that solves valuable problems for you, right? And those problems and those bottlenecks are going to be different for everyone. So breaking this down a little bit further in terms of how we see automation, automation is a set of tools, both hardware and software, combined with processes that enable, enhance, and accelerate life science research. That's divided into a couple of different groupings or types of automation. I think first, most people are familiar with task-based automation. In task-based automation, you're really looking at the alternative from, do I do something manually versus do I get a tool that can do it for me? And then the kind of, in our mind, we can kind of have workflow-based automation, right? These are systems that we've seen in labs that sit on a benchtop, usually consisting of a couple of tools, maybe as many as half a dozen tools working in concert. But that's not always the case. Sometimes in workflow automation, you can have a very full-featured device, as liquid handler as an example, that can meet some of the needs of task-based and workflow-based automation. And then finally extending to process automation, right? And process automation is where you're really looking at the objectives of the research from start to finish and how you organize not just the automation, but the data and the user processes that are occurring in the lab. When we look at automation here, I think it's important to assign kind of a key verb to each of these. In task-based automation, we're focused on enabling. So sometimes in task-based automation, we're enabling something that users cannot do. An example of that would be in flow cytometry, right? A user could operate an acquisition device and write down a single data point, but it's not really feasible for a user to record data points over time at the rate that you can get out of a lot of this instrumentation, right? So just by looking at a lot of the instruments in our industry, you're actually hitting most of the low-hanging fruit in automation, right? That's where the biggest gains are because you're enabling something that's not able to be done. In workflow-based automation, we're typically focused on enhancing. We're not trying to add a new capability, right? You really want to focus on capability development in task automation, whereas bringing things together is workflow automation. And then when we look at process automation, we're really not concerned with the tools at all, right? We're more focused on what's happening in the lab, whether it's the users, the data, the labware, other teams, customer samples coming in, right? You're focused a lot more on that than you are on the actual hardware being used in the automation itself. All right, so with that context, why don't we move into some of the tools and thought processes that we can use to put the automation in practice. Going back to what we're trying to do in labs, this is an example research process used for T-cell therapeutic development. All right, so this is clearly high level, abstract, but comprehensive. Right? When you're looking at what's happening in the lab, all of these things need to occur. And when you have somebody that's involved in developing the research, but not really in touch with the automation, there's a gap to say, how do we take this and how do we start bringing automation in? And how do we do it efficiently and intelligently rather than just throwing a lot of hardware or dollars at it without a lot of gain? So looking at solving this problem, there's kind of two main ways that you can approach it. We've used the terminology inside out versus outside in. Some people would call this bottoms up versus top down. So if we look at inside out, right, we're starting with a task, with some low level task that we're trying to accomplish, and then we start to string those steps together, right? Bottoms up would start with looking at your SOPs, right, your benchtop SOPs, and say, we're going to start figuring out what the pieces are, right, and string them together, and hopefully we end up with something that works as a complete solution. Bottoms down, or sorry, top down automation, or outside in would be where you look at that main workflow that was on the prior one. You start breaking it up into chunks, right? Because that already kind of had some of the work done by being broken down into high-level module steps. And then you really decompose that down. And once you get to those more granular views on things, that's when you start looking at hardware. What we found is neither of those are right, and neither of them are wrong. It actually takes both views, right? So ideally, when you're working through a process of automating research, you want to start top down. You really want to break it up into logical steps that have key bottlenecks in the lab, right? Or logical steps where within an individual area, you have certain technical needs that are associated with it, whether it's temperature control, environmental control, or sometimes if there's incubations or out-of-house processes, right? There's a lot of drivers that will tell you how you want to break up that process from an automation standpoint. That's really architecting, right? So we want to architect top down, but then when we're rolling out automation, we really want to start bottom up, and that's looking at the actual specific technical needs and finding the right tool for the job, rather than focusing on the big picture. And really, when you do those together, you meet in the middle where you have the right hardware from a bottoms-up approach, and the right architecture from a top-down approach, and then you use the tools that are available within our industry to link those together to give you a comprehensive automation solution. The main finding here is that automation is always modular. A lot of people will walk into a lab and they'll look at a table, they'll look at a big system, and they think of that as an automated system. And that is entirely true, right? At the high level, when you're doing process-based automation or workflows-based automation, you do want to have the ability to step back and look at it and say that this does this process from start to finish. But the reality is, in rolling out any of these systems at any scale, they are always built modularly. And you need to keep that in mind, because that has impact both in terms of how you build these systems, it's how you maintain these systems, it's the day-to-day operational impact, right? That you end up having a lot of modules, so there's a bit of a learning curve associated with them. But one of the main huge drivers of value in this is that allows you to build these systems a piece at a time, right? And address your highest value needs first. So when deciding what to automate, you really want to ask yourself, what problems are we trying to solve? You don't need to solve every problem at once, right? You identify the critical problems, you put targeted modular solutions in place to solve those problems. What breaks if this step fails? This is a key one, right? If you have a solution that automates start to finish and something happens in the middle, now you have a long continuous process with no ability for error resolution, right? You want to look at what tools you have in your organization, right? What support you have, whether it's support of other team members or support from a facility standpoint. And then you really want to think about if it scales, right? It's not just about what you're trying to automate today. It's about that next step, right? So thinking about what does this look like as I design my lab in a modular fashion. So in putting a lot of these automation solutions in place, there's some pitfalls that we've seen. And I want to share a few of those with you and try to use that as a way to give you some ideas on how to approach automation in a more efficient way. One of the key ones here is over automation, right? And this really gets down to that architecting of a solution in a logical way and being deliberate on where you place automation rather than just saying we're going to automate. You really want to make sure you put it in the right place. And the reason that this becomes a pitfall is there's a trade-off always between usability and flexibility, or convenience and flexibility. When you look at low-level tools, instruments, generally those instruments have the greatest degree of flexibility, and they're easy to use, right? But they're not necessarily scaled up. And as you build more automation, as your degree of automation goes up, you end up automating more of your workflows and more of your processes, right? That the impact of that is as you automate those workflows and processes, you are now tied to those workflows and processes. So that has ultimately an impact on your day-to-day usage, which might be okay, right? And that's why when you look at something like industrial automation, a little bit outside of our space, you see higher degrees of automation because those processes are far more rigid, far more fixed, and the desire for flexibility is less. We are doing research, so the ability to change what we're doing is important, right? Doing the same thing over and over again is not going to generate new results. So that thinking about this trade-off when you're rolling out automation, it drives you towards deliberate automation, but also towards more smaller modular automation, right? Because that gives you a higher degree of reuse. So rather than thinking about everything as process automation, you really want to focus on the tools, the workflows, how you can reuse those because those help you maintain flexibility in your lab. And ultimately, you can bring those together for a higher degree of automation, but that's not always the right step. Another huge pitfall that we see is Swiss Army knife automation, right? For those unfamiliar with the analogy here, a Swiss Army knife is a multi-purpose tool, right? Get an image of it on the right where you can basically do everything with it. So that does sound appealing to labs, right? Is you're going out and you're going to make an investment, and this is probably the biggest investment that you're going to make in the next several years. And you want to make sure that you get it right and you get the most value out of it, right? So this is going to the prior analogy. This is saying, I want flexibility, right? But the end result of this is that if you do that at scale, you end up with systems that really just don't work, right? Because it's impossible to design a large scaled up system that does absolutely everything, right? So when you look at this as if you want that high degree of flexibility, you want to be able to repurpose it, deliver it in a bunch of different workflows, you really want to be sticking more with task-based automation. In the workflow automation arena, right, you can imagine some of your research processes that are generic enough that you can reuse them for a bunch of different research needs, right? And that's not always a problem because you can do that. But basically, the more fixed purpose that you want to be on a system, it is impossible to support that if you want to do 10 other things on the system, right? So understanding that trade-off and making sure that you're well situated on it is important. This is one that never really comes out on the front end of automation, but ultimately, if you look at the success of automation 6 months, a year, 2 years down the road, usually the reason that these systems do not get the value or the ROI that our users are looking for is because of a lack of awareness of what it takes to support automation in your lab. So what we have here are a bunch of different roles, and this is from an idealistic, let's say, comprehensive automation. These are all the roles that you have to have internally to be successful. Now, sometimes one individual might wear multiple hats, but usually you're really just looking at it as a scientist or sometimes as a lab manager, right? But to be successful, you have to put all these different hats on. This doesn't matter as much when you're going to buy instruments, right? But as you scale automation, this becomes fundamental. And that's not just in the post-installation phase when you're deployed. This is critical in the design phase when you're saying, what is this? What automation is appropriate for us? These are all stakeholders that need to be involved in the architecting of that automation solution. Even if you're looking at workflow automation, as soon as you start talking about data that has to move between labs, all of these stakeholders in your organization have to be involved in the beginning. And you need to really think about what is the long-term impact for them, because if you don't have buy-in, it doesn't matter if you get the budget. If you don't have buy-in for the other people to interact with the automation system, not necessarily directly, but by providing work for it or analyzing results from it, right? There's a lot of reasons that you need all of these other individuals to buy in, or automation will not be successful. And their buy-in will help you architect a far more effective solution for your lab within the context of the research that you're doing and the organizational support that you have. And this one I had to throw in because this is a bit of a pet peeve of mine. We do talk to users where we ask them if they're using barcodes, and they wonder why, right? Because we can just write on a marker on the plate, right? Or we don't have enough scale right now. When you look at automation at any scale, you look at data, you look at connectivity, any of these connected solutions, you need to be able to link results together, right? And this isn't just about compliant traceability. This is just about putting systems in place that allow you to have confidence in your data. So I would encourage everybody, regardless of scale, you need to think about how you're going to put these modules together, and you have to put a system in place. So barcoding is by far our strongest recommendation. Just do it. Doesn't matter if you have 1 instrument, 2 instruments, 5 instruments. This allows you to scale by putting those processes in place from day one, even at manual scale. You build that in, and then you can build it up. So last, I kind of want to dive into, dive is maybe the wrong word, give you a very brief overview of some of the tools available for automation, because obviously this is an extensive toolkit that's available throughout our industry. When looking at some of the solutions that exist for this, looking at task-based workflow and process-based automation, we have a variety of solutions, right? We think mainly about hardware, right? Automation, going back to kind of where I started on what do people think of automation, usually people think of robots. And that's a very hardware-centric view of automation, which is perfectly valid and accurate when you're talking about task-based automation. That does scale a little bit into workflow automation. A great example of that would be liquid handlers, right? In liquid handlers, you're not just doing a singular task. Usually you're trying to take advantage of the capabilities of that instrument to link together a bunch of sample prep tasks, right? And sometimes liquid handlers are bringing in deck accessories that allow you to do more than just liquid handling. So that is an example of vendors and manufacturers in our industry that are building tools that allow you to scale without having to make further investments, right? That doesn't mean that you avoid all the work that we've been talking about before, right? Because that workflow automation, you still have to break down the problem correctly. You still have to have the right organizational support. Kind of the next phase is looking at, I'll call it integration software, which software is maybe a little bit of a misnomer here, because there is equipment that's designed specifically for workflow-based automation, right? When we think about robotic arms or automated incubators, there's components that really by themselves, they don't do a singular task that well, but they are fundamental for supporting workflow-based automation. But kind of the key is that workflow-based automation has tools, both hardware and software, that are designed for that. Now, when we look at process automation, similarly, there's tools that are designed specifically for that. So one of I think the main point of emphasis that I want to get across here is that when you look at automation scale across all these different scales, you have different tools that are appropriate for solving different problems, and you do not want to try to solve every problem with the same tool. That ultimately actually is a different pitfall, right? When we're looking at buying an instrument, and all of a sudden in that instrument we're trying to do task-based workflow and process-based automation on the same instrument, it's not the right tool for the job, right? Can you force fit it? Maybe, depending on the vendor, but that really isn't going to get you a successful deployment in your lab. So a couple of things to look for in tools, right? In tools, we're talking about individual instruments here. If you ignore every bullet point on this list, most important is, does it meet the technical requirements, right? When we think about scaled up automation, everybody's thinking big picture, and this is, again, kind of that trade-off between looking at process automation versus tool-based automation. It doesn't matter how tall you build the building if your foundation is wrong. So when you're in tools, you really want to make sure that you are finding the right tools to meet the technical objectives. Even if it is not an ideal automatable solution, with the right partner, you likely can still automate it. Obviously, it would be great to have the right tool with the right automation support, right? And that's kind of some of these later ones. Can it meet the technical requirements of tomorrow, right? Does it have the extensibility? Does it have built-in workflow support, right? Are you going to have the ability to turn it from just a single purpose-based tool to do a little bit more of the workflow? And there's vendors that are adding technical capabilities that bring built-in workflow support to this. A good example would be, fluorescent reader right now. Rather than just doing reading, we've got readers available that do injection of small amounts of reagents for kinetic assays, right? So that typically would have been a workflow, right, where you had to do a dispenser combined with a data acquisition device and pair those together with some other automation. But vendors are driving that towards giving you the ability to do workflow-based automation within a single platform. And then some of the, I'd say to me more obvious ones, but maybe not for you, are you need to have APIs, right? API, for those unfamiliar with, is the application programming interface. That's what allows software to talk to other software. So when you look at building workflow-based automation, when you're putting that workflow together, the individual tools that you select that might be the right tool for a job have to have those software hooks that allow the workflow automation to happen. And then from a hardware support, this is a little bit more complicated than just saying, can you pick up a plate, right? So the diversity of how you pick up a plate, like does it support a variety of labware grippers so you're not pigeonholed into one vendor? But also, where is that plate presented, right? So you might look at an instrument that works great standalone as a benchtop instrument, but just because of how it presents the plate, if you want to do any workflow-based automation with it, it totally eliminates the ability to do manual usage on the instrument, right? Just from a physical access standpoint. And that's not even talking about the scale of automation. It's just saying physically you can't reach it. And also footprint and layout, is kind of tied in with that. What to look for in workflow automation? So workflow-based automation, as I mentioned before, is primarily driven by software. You want wide support for tools, right? So that there's a number of vendors out there that have wide support. You want the ability to flex a bit to process automation, right? So that means whether there's some inherent data handling capabilities or orchestration capabilities, or there's the ability to integrate with LIMS systems. But probably importantly here is the partner starts to come into play. When you're looking at tools, you're focused on technology. And because you're going to be using it and you have the power as the user. When you look at workflow-based automation, configuration starts to become critical, right? And that configuration means that you need somebody to assist with that because you don't have the support of in-house automation engineers that are going to do all the configuration for you in most organizations today. And then when we look at process automation, I want you to focus first on the shape of the diagram on the right. This is a pyramid, right, which is tools configuration and data and SOP or users would kind of be in that top tier. This is different than the some of the depictions that we had earlier in the webinar, and that's intentional because this is wrong. When you move to process automation, tools are almost negligible. The hardware is negligible. You really spend all of your time focusing on processes, and those aren't just, it's not just the scientific process. I've used the word user automation before as kind of a way of describing what process automation looks like. Is you're linking together all these modules and steps, and you need to tell people when they need to interact with the plates, when they need to load consumables, when they need to do air resolution. And all of those concerns start to become not really focused on the science, but focused on what's happening in your lab, right? And that's a decision trees that are not associated with the core assay. So what you really don't want to have is, as you scale up automation, you don't want to spend all of your time focused on the hardware. You want to spend you want to be in a spot where the hardware has already been validated. You know what tools you're going to use, you know what tools work for your science, and you focus on configuring those tools and then effective use of those tools in your lab, which is more focused on the data and your user processes. And definitely when you get to this level of automation, the partner is critical. So it's partner, it's organizational support, it's all of those things really drive what's happening. And now I'm going to turn it over to Richard to give you a deep dive into the tools that Bio-Rad has built for task automation and the extensibility for scale.
Thanks, Andrew, and thank you to Hudson Lab Automation for allowing me to be part of this presentation. Collaborating with them on this webinar is symbolic in a way because we see laboratory automation as partnering between instrument suppliers, integrators, and researchers to get the best possible solution. So I'm going to talk about automation in flow cytometry and gene expression analysis, starting off with flow. Now, a large number of our customers that are interested in automating flow cytometry are involved in screening for drug discovery, and it tends to fall into one of two categories. The first is in the hit discovery phase, and this is sometimes called primary screening. Here we have binding assays and phenotypic screening. In both these cases, it's really the volume of samples and the time to result that our customers are really interested in. But I just want to echo the sentiments that we heard earlier. It's not really about speed as much as utilization of the available capacity. Binding assays are common in antibody discovery. Here you have a target expressed on a bead on cell surface, and you look for binding of a labeled candidate to that target. The advantage of using flow cytometry for this is not only does it facilitate multiplexing, allowing you to look at multiple things simultaneously, but your target is expressed on if your target is expressed on the cell surface in vivo, then it's in its natural context. It will be folded correctly. The protein will be folded as it is in vivo, and that reduces the chances of false positives as well as false negatives. Phenotypic screening is more commonly used for small molecule discovery. Here we're taking cells and exposing them to a library of compounds and looking for phenotypic changes in those cells. Now that could be apoptosis, proliferation, activation, or even cytokine secretion. The point is we're looking for a change to the behavior of those cells rather than a binding interaction. And of course, these compound libraries can be huge, so automation makes perfect sense here. In both cases, another benefit comes from the improved data handling. With the sheer volume of data that can be generated, it's a huge advantage to be able to automate processes that export, store, and catalog data. Next, let's talk about secondary screening. This is typically part of the hit optimization stage of drug discovery. We already have a candidate or a short list of candidates, and we want to test how they perform or are likely to behave in vivo. There are many different assays that can be performed. This is not a complete list. And I'm just going to call out two, which are apoptosis or cell killing assays and immunogenicity assays, because these two are quite common. A lot of drugs have a mode of action that relies on selectively killing one type of cell whilst not affecting other cell types. Here we have a rituximab biosimilar that selectively targets B cells. And you can see we get a significant drop in the proportion of B cells expressed in this CD3 negative, CD19 positive phenotype. And although this looks like a pretty simple figure, there are more than 80 data points on this. So you can see how this type of experiment quickly becomes time consuming, especially if we want to assay multiple drugs against cells from multiple donors. Also, when we get to this phase of drug discovery, regulation is starting to become increasingly important. And that's another thing that automation can really help with. That's also the case for immunogenicity assays, where we may be looking for the activation of a set or subset of immune cells caused by the presence of the drug. Now, generally speaking, activation means that the drug will be mopped up by the immune system and stop working. Quite often this will be performed on dozens, if not hundreds of donor samples. And just as in the apoptosis example, we're likely looking at a specific subset of cells, usually B or T cells, from a mixed population. And this is why flow cytometry is so useful for this assay, because it's really the gold standard technology for identifying and counting cells. Now, all these types of screening assay are very well suited to Bio-Rad ZE5 cell analyzer because it combines a really high event rate, which is especially important in assays with multiple cell types, with fast plate handling, powerful optics, and of course, a host of automation-friendly features. So what to automate? I've already mentioned some of the applications. Let's talk about what parts of the workflow should be considered for automation. We've integrated our instruments in very simple work cells all the way up to very complex multi-reader setups. It's important to think about which stage of the workflow is going to be give the most benefit from automation, and spend some time thinking about the workflow and where the bottlenecks are. Each step is going to provide some level of benefit, but of course you have to weigh that against the cost. In the example above, we see a simple reader-feeder system. And to give you an example of the increase in throughput that you could expect, we're talking about a threefold increase from just this step alone. Now, using the ZE5, that's a difference of about 6,000 more samples you could get through in a day. Next, you might want to consider some form of temperature control for sample storage. After that, you may find that sample preparation is now holding you back in terms of throughput. The bottleneck has kind of shifted to that sample prep stage. So considering a liquid handler upstream of your reader will help you improve your throughput, but it will also improve your assay reproducibility. And obviously help you to cope with those extra 6,000 more samples your reader is now capable of handling. So I've talked a lot about flow cytometry applications. Let's have a look at qPCR. Pathogen detection is a critical process in food safety, clinical diagnostics, and environmental monitoring. Laboratories face increasing pressure to deliver rapid, accurate results while handling large sample volumes and meeting stringent laboratory regulation requirements. Now, traditional manual workflows for pathogen detection involve multiple steps, including sample enrichment, DNA extraction, PCR plate setup, and real-time amplification. These steps are labor intensive, they're prone to human error, and they're difficult to scale during periods of high demand, such as an outbreak investigation. Automation addresses these challenges by streamlining the entire workflow, reducing variability, and ensuring compliance with regulatory standards. Bio-Rad's IQ Check real-time PCR solution, combined with the CFX Opus Deepwell real-time PCR system, offer a validated and integrated approach for detecting pathogens such as Salmonella, Listeria, E. coli, and STEC virulence genes. By incorporating automation, labs can achieve faster turnaround times, improved reproducibility, and enhanced data integrity. When considering integrating any instrument for part of your workflow, there are certain key questions that you need to think about to assess how well that instrument is likely to perform. A big one is the loader design. Is it easy for a robot arm to access the loader? Here you can see the loader from the PTC Tempo, which is an automation-friendly thermocycler with a really accessible loader. Next, does the instrument have automatic fault detection and recovery? Now, every instrument will occasionally throw an error, and if there's an automatic way of correcting errors, that means that your run doesn't end and the system can recover. Now, this is something that we built into the ZE5 cell analyzer, which has a feature that detects blockages and stops acquisition before it gets too bad. It can then clean the sample line. Does the system offer automatic maintenance features? Now, this is not so much of an issue with qPCR, since there's very little day-to-day maintenance required. But flow cytometry systems need daily cleaning, usually prior to shutting down. If cleaning requires manual steps, that's going to be a problem if you expect your run to end at 3 a.m. on Saturday morning. Again, this is a feature that is included as standard with the ZE5 cell analyzer. And lastly, it's really important to assess the quality of the API library. I think this is something that researchers really need to ask their instrument suppliers about, because it doesn't matter how many features of a system, how many features a system has, if those features are not available through the API. They won't work with automation. And this is something that we have paid particular attention to at Bio-Rad to ensure that our customers can use automation to get the most out of our instruments. So in summary, qPCR and flow cytometry are examples of discrete workflow steps. As we've seen, automation may start with a simple plate loading, and workflows can expand to include incubation, sample preparation, and analysis. And of course, we design our devices to seamlessly integrate with automation. So I've spoken about some of the tools we've developed at Bio-Rad. I'm going to pass back to Sarah, who will talk about how Hudson has built up tools to support workflow automation.
Thank you, Richard and Andrew. I'm going to walk you through a few real-world automation examples to make this concrete. Before we jump into the examples, I want to quickly orientate you to the Hudson platform, because everything you're about to see is built on this same foundation. At the platform level, Hudson combines three core elements. First, workflow orchestration through soft links. This is the software layer that schedules, coordinates, and monitors every step in a workflow. It abstracts individual devices, so you're managing a scientific process end to end, not instrument to instrument handoffs. Second, robotics and plate movement infrastructure. This includes robotic arms, plate storage, tracks, and transport that moves plates reliably and repeatably between instruments. Third, deep integration and application expertise. Our engineers have integrated over 300 analytical, sample prep and storage instruments, along with pre-configured workflow templates for applications like flow cytometry, ELISA, colony picking, and bioprocessing workflows. A key takeaway is that this is a modular platform. Customers can start small and expand without re-engineering their system. So the first example that I'm going to quickly talk about is a label-free integration. This is very simple and that's intentional. This is plate loading automation for an antibody therapeutics company using the Gator Bio label-free BLI technology. In this workflow, the BLI assay requires a 10-minute incubation inside the instrument. For this particular customer, that incubation time was a bottleneck. In addition, the plate loading was a bottleneck for them. So in this example, we automated plate loading to and from the instrument and also added external heated nests so that the incubation could start outside the instrument. By the time the plate reaches the BLI system, the incubation is already underway, which reduces this idle time and improves overall utilization. This is a great example of automation delivering value by removing dead time and incorporating automated plate loading. The second example shown here is more of a workflow automation, and this enables microbiome discovery. This is a full workstation operating inside an anaerobic enclosure for translational research using human fecal samples. In this system, SoftLinks orchestrates 18 distinct devices, including plate sealing and resealing, dispensing, colony picking, and imaging. All of these steps are coordinated in a single workflow. Equally important in this particular example is what happens after the imaging. So images which are generated on the Hudson Rapid Picker are automatically routed into a downstream AI pipeline, which supports the identification of low abundance microbial species and feeds those decisions back into the system for targeted colony selection. This work was done in collaboration with Columbia University and also published in Nature Biotechnology. The results speak for themselves. Over 27,000 isolates spanning 80% of the abundant gut species. More than 100,000 colony images used for phenotype-driven selection, and direct linkage of morphology to genomics. What once took weeks of manual labor is now completed in days, really enabling high diversity microbiome biobanks and unlocking new sites into host microbe interactions. This automation is it's not just scaling science, but it's changing what science is possible. In this last example, this is a toxicity screening example, and this highlights one of the more demanding workflow automation challenges that we see in labs. Unlike many assays, whole organism screening is biologically fragile and extremely time sensitive. This whole workflow takes 5 to 7 days. Once the embryos are prepared, there's a narrow window for dosing, incubation, and imaging. Missing that window means experiments fail and the data is lost. In this case, automation is not just about plate handling, it's about orchestrating a multi-day end-to-end workflow where every step happens at the right time, in the right order, and under the right conditions. So this particular system that you can see here coordinates picoliter level chemical dosing, temperature-controlled incubation, multi-imaging systems across different modalities, barcode tracking, and data associations, and then the export of results into analytics and LIMS environments. The real value of this workflow is that it allows researchers to screen large chemical libraries, study developmental toxicity, neurobehavioral effects, and antiviral activity. In the context of this webinar, this example illustrates how workflow automation enables biologically complex science at scale while protecting data integrity and dramatically reducing labor and variability. So the case study takeaway really across all three examples, the pattern is consistent. Automation is modular, value is context dependent, and the right platform allows systems to evolve without re-engineering. Automation is not manual versus lights out. It's about identifying the steps that matter the most, automating it well, and ensuring the system can grow as scientific needs change. And with that, we'll move right into our live Q&A. Please continue submitting questions through the Q&A panel, and we'll address as many as we can live with Andrew and Richard. Thank you. Hello, and thank you everybody for joining us today. So we're happy to be back live with you. Right now you'll see a short survey pop up on your screen, so please feel free to answer those questions that come up, and otherwise they'll disappear after 30 seconds. Yeah, and please continue to add your live questions for our experts by clicking on the question mark on the icon on your screen. We'll answer as many as we can, and for those that we don't get to, we'll follow up with you directly post-webinar. Okay, so I'm going to ask the first question that I see, and this one's for Richard. What plate formats is the Z5 cell analyzer compatible with in an automated environment?
Yeah, so it's got fairly broad compatibility, so it can handle pretty much any plate format. But obviously, in an automated environment, you're talking about the the high density plates, so 384 well plates and 96 well plates. But kind of any format of those two. So like round bottom, flat bottom, V bottom, and from any manufacturer as well. So you've really got you can use your favorite plate. Obviously, it's important to ensure that you have a plate that is compatible with automation as well. So it's fairly rigid, so it's gripped well. But yeah, you can pretty much use anything that you want.
Thank you, Richard. The next one is for Andrew. Where do most labs often struggle when starting automation? Is it choosing what to automate first, or really how far to go?
Yeah, I think both of those are important. Really the big struggle for people is automation. Again, it's that big picture automation, right? And the reality is that's not something you can't do everything at once, right? So I think it is about deciding what to do first, right? Really narrowing it down to what are those key bottlenecks. How do I solve those problems and provide immediate value, and then build that over time?
Thank you. The next one is how does Hudson integrate systems with existing LIMS and ELN systems in a regulated lab environment? I think that one's for you, Andrew.
Yeah, that brings up pretty a broad set of concerns, right? And when you start talking about compliance and data spanning multiple systems, so we have tools built into SoftLinks to allow it to work with instruments first and foremost, right? The device is generating the system to try to organize that data, and certainly if you're using barcodes, try to correlate that data between multiple different instruments and devices. And then, interacting with LIMS systems, we've worked with most of the major LIMS systems on the market. We have tie-in and hooks, but I don't want to undersell the effort required to do that, right? As I think all of you are aware, when you use ELNs or LIMS, you really have to spend time tailoring the data structures and the schemas to your processes. So then within SoftLinks, you basically have to teach it that same verbiage and nomenclature that you use in your lab, right? So we have the key software hooks out of the box, and then through configuration, you can teach it how you work in your lab.
Perfect, thank you. Another one for you, Andrew. So you've shown a robotic arm automating plate loading in front of an instrument. Does one arm typically map to one instrument, or can a single arm serve multiple instruments, such as 2 or 3 flow cytometers, and how do you architect that?
Yeah, the answer there really comes down to what problem are we trying to solve, right? If we're talking about flow cytometers as an example, usually you're talking about pretty long reads, right? Because some of the biggest gains in flow are going from not having an auto sampler to having an auto sampler, right? And when we're talking about an instrument like the ZE5, we're talking about a very capable instrument that is much different than what was available 10 or 15 years ago. You can read 96 samples at the same time, totally hands off, and that might take you, depending on the flow cytometer you're talking about, anywhere from tens of minutes to many hours. So when you have an instrument like that, absolutely, one robotic arm makes a ton of sense, right? You can service a bunch because you're just trying to solve the problem of keeping those instruments busy when somebody isn't there. If you're trying to solve a problem where you have very precise timing constraints, sometimes sharing a robotic arm is not the right answer, right? Because it becomes, from a flexibility standpoint, if you want to do a couple of different things on the system and you want to have very tight timing constraints, you may not be able to hit both of those requirements at the same time with one arm.
Thank you. Next one's for you, Richard. Does Bio-Rad offer pre-mixed antibody cocktails for flow, and how do those fit into automated workflows?
Yeah, so antibody cocktails is one of the kind of things that that are quite useful for things like screening assays, because it means that you can you can make up your assay once and just reuse the cocktail. So it really fits in well when you've got lots and lots of samples. We don't actually offer pre-mixed cocktails. The reason behind that is because we find that there's just too much variation in terms of what our customers want to do. But we have recently, well, over the last couple of years, we've been releasing a range of dyes that we call Starbright dyes, and these dyes are really amenable to pre-mixing. So we've pre-mixed cocktails up to like a year in advance now, and we see no difference. And so what that means is that our customers can, once they've decided what antibodies they want to use, they can make up their mix once and use that for at least 12 months.
Thank you. Next one for Andrew. How long does it typically take to design, install, and commission a system like the workflows shown today, from initial concept to running experiments?
Yeah, obviously, if you're looking at getting your foot in the door with some task-based automation, you can get instruments in the scale of days to weeks. If you're talking about moving that to the next step, where you're just going to try to load some plates into it, right? You're trying to make more effective use of that instrument. That's usually a pretty close to out-of-the-box project that could be done anywhere from weeks to months. But when you really start looking at workflow, extensive workflow automation and process automation, that's going to vary dramatically based on the scope of what you're trying to automate. Because you spend a lot of time interacting with, doing process design, process development, interacting with stakeholders in your organization, ideally not trying to redesign your process around new labware, even though that does happen at times. So, for end-to-end automation in a workflow like that, the one that we showed, the T-cell therapeutic, that could conceivably be a multi-year project to get eventually all of the automation pieces in place to meet everything that you anticipate doing on that workflow. But the average, you just got to give everybody a benchmark on this. The average we see for small to mid-size automation is kind of like a 2 to 6 month time frame, depending on scale, depending on whether you have the instruments already or not.
Okay, we're on the hour now, so for the remaining questions that we have, we'll follow up with you all directly. And at this point, we're going to close the webinar. And thanks again to Richard, thanks Andrew, and thank you for all of the people that attended, and we'll follow up with you. Thank you very much.
Thank you.
Thanks, everyone.